Hello. Am I audible?
Yes, sir.
Yes, sir.
Okay. So, is there any feedback noise or anything from microphone?
No, sir.
No. Okay. So, what we have discussed in the last class?
We used to identify it assay formats, sir.
So, we completed biochemical and cell line assays, right?
Yes, sir.
Differences between biochemical and cell line assays, right?
Yes, sir.
Okay. So, last class, I think if you remember, I called two number. One is 122013004 Aparna and 3003 Aparna. So, any of you present today? So Aparna Venkatakrishnan, your net is slow, right? Still, you can unmute your microphone. I think I called you people both in the last class. I don't know where you people went. Can you people unmute? I have some question for both Aparna. Can you unmute? Okay, I don't know what's happening. Am I connected? Am I audible to others?
Yes, sir.
Yeah, okay, fine. So let me proceed, right? Is my screen visible? No, okay. So is it visible now?
Yes, sir.
Yes, sir.
Yes, sir.
Yes. Okay. Okay. Good.
Yes, sir.
So far our knowledge is we have some idea about what is meant by drug. Also we discussed about drug targets. So we studied about the 9 steps of the modern drug discovery process. Okay. So the first step is target identification. Second step is hit identification. The third step is hit to lead. The fourth step is, what's the fourth step?
Lead optimization.
Lead optimization.
Okay, lead optimization. Then next step is
IND enable.
IND enable. Okay, so this is what we have studied so far, right? Also we discussed about ADME, absorption, distribution, metabolism, excretion. That is, the journey of the drug molecule within the human body. So we know the difference between the hit molecule and the lead molecule as of now. We know the definition for hit molecule and lead molecule. So we know the definition for lead optimization. Again, I just wanted to start with this lead optimization. I'll cover the IND enabled process and the clinical studies in this lecture. Okay. So any question, you can stop me at any time, right? So no need to wait till the end of the lecture, right? So if you have any doubt, just stop me, right? So I'll address the questions. So lead optimization is the process by which the medicinal chemist will make small changes in the lead molecule to make it a better molecule, right? So so during the process of preclinical studies, so this IND enable and this this IND enable is just after the preclinical studies. Okay. So during this hit to lead and lead optimization process, so we have to start a stage that is called as preclinical studies. That stage is called as preclinical studies. So the main idea of preclinical study is the molecule that we have has to be evaluated on animals. So why normally we evaluate the molecule on animals? Because we cannot directly try the molecule by giving to humans. Obviously, we design the drug molecule to treat some diseases related to human mostly. So, but we cannot directly give the molecule, and we cannot directly check the effect of the molecules on humans. That's not possible. So the main reason for this is, because the molecule may have toxic effect on humans. We cannot directly evaluate on human. So to avoid that situation, we choose some animals and we evaluate the molecule first on animals. So once if the molecule is very safe, if the molecule has good efficacy on animal, then we take that molecule further to the human. Okay, so the first stage of this study is called as a preclinical study. Once if the molecule is successful on preclinical study, then we can take that molecule to human system. We can carry out the clinical studies. So the main difference between preclinical and clinical study is, in case of preclinical study, the study is done using some animal models. In case of clinical studies, the immediately after preclinical, the study is done on humans. That is the difference between preclinical and clinical studies. So we don't have any stages on preclinical studies, okay, but we have 4 stages on clinical studies, like phase 1 clinical studies, phase 2 clinical study, phase 3 clinical study, and phase 4 clinical studies. So clinical studies, we have 4 phases. We don't have any such phases on preclinical studies. Okay. So so preclinical studies, maybe for next 10-15 minutes I'll be talking completely about the preclinical studies, what normally we do in preclinical studies, right? So in preclinical studies, we have to use the animals as model. Okay. So because we cannot directly give the drug molecule to the human, we have to evaluate that using animal models. I just write it as animal models. So if we say animal, it's a very general term, right? So there are many organisms which comes under animals. So what are the animals we can choose for preclinical studies? Different animals we can choose. I just wanted to give a few example of animals which are well used for this preclinical studies. Say we can use zebrafish. We can use xenophus frog for your study. So this is a fish for your study as an animal model, or you can use mouse or rat. Okay, those comes under certain category. We can use rat, mouse. They're excellent, excellent species. As animal model, or you can also use primate like monkey, dog, and all. those also can be used, right? So how do we choose this animal for animal studies? That depends on the type of disease in which the company is working on. Okay, so for all the diseases, we cannot use randomly this animal. Specific animals can be used for specific studies. That's the idea, right? So I just wanted to give some examples. Say, for example, if you are working for a company and you are working to discover some drug molecule which is related to the developmental biology. So if your intention is studying developmental biology, if your intention is studying about the developmental biology, then the best preferred model for that is zebrafish. Okay, so so if you are working with a company and your intention is studying some tissue induction, some of the terms may be very new for you, no need to worry about that, right? Just follow me, right? So I'll make you to understand everything. So if you are going to do some experiments related to tissue induction, so that time we don't prefer for the zebrafish or any other model. We always prefer for xenophus. Sorry. So we just go with frog, right? So we use the frog as the animal model. And if you are working with if your study is anyway related to organ function, or say if you want to study the function of heart, organ function, heart is an organ, right? Or if you want to do some studies on tumor xenograft, so the preferred model is mouse or rat. We can go with those, right? So those are very good for studying the organ function and tumor. Those are excellent animal models. So if your intention is studying about some organ physiology, the best way to go with is you can go for the higher animals like monkeys or dog. Those are highly preferred. So what I'm trying to say is we cannot use randomly these animal models. The way to choose the animal model for your preclinical study, that depends on different factors. Okay, so if you are interested, if you want to study if you want to design a drug molecule for diabetes, the best option is going for mouse or rat. So if you want to study about Alzheimer's or Parkinson disease, we just go for monkeys or dog. Those are the very preferred model. So so far I said so many points. The take home message is that how to choose the animal model is that, so whatever disease you are working on, okay, so whatever you want to understand about the disease, that should more or less exactly mimic by the animal that you are using. That's the point, right? I cannot mimic the human nervous system brain system with the brain system which is present in the mouse, but I can somewhat compare the brain functioning of the brain and the nervous system which is on monkey or which is on dog with the human system. So that's why we prefer the monkey or dog for Alzheimer or Parkinson diseases. Okay, so and also sometimes we have to ensure that the metabolic pathway that we are trying to work on, that is exactly or almost similar to the human system. The animal you are choosing should have a very close resemblance with the human system. So that is how we choose the animal model for the experiments, right? You may have some doubt at this point. If any doubt, I can clarify it. So any doubt?
No, sir.
Any questions? No questions. Okay. Hope you are following me, right? So if no questions, well. So what we normally do is, say for example, let's say that your company is working on type 2 diabetes, right? So you want to discover a drug molecule for type 2 diabetes. That's the intention of your drug discovery project. So in the biochemical assays and in the cell line assays, initial screening, these are initial screening, right? So you got some hit molecules and lead molecule, so which you assume that they are promising on the biochemical and cell line assays, right? So now you wanted to evaluate this molecule to the human system, but very unfortunately we cannot directly do that. So you have to perform now the preclinical studies. So while looking for the animal model, you have different options, but you are picking say you are picking the rat as the animal model, most preferred animal model to study the type 2 diabetes and type 1 diabetes. So you are choosing the rat as the animal model. You are very confident that the disease that is in rat and in human type 1 or type 2 diabetes, they are comparable. So you are choosing rat as the model, right? So initially, for working with preclinical studies, right? So we cannot directly use, normal rats. We have some specific laboratory rats and mouses. You have to choose only that. So there are some specific breeds there that alone you can use for your research. You cannot randomly pick up, the outside rat or mouse that you see in your house, right? We cannot use them. So you have to get approval first to working with the animal models. Approval is very important for working with animal models for purchasing that specific breed, right? So who is going to give the approval for the study? So every institute, right? So will be having animal ethical committee, institutional animal ethical committee, IAEC, right? So stands for institutional animal ethical committee, and you have to submit a proposal to institutional animal ethical committee stating that which animal you are going to use, what for you are going to use, and how you will be using that animal for your studies, and how many number of animals you will be using for your experiment. So those informations should be submitted to institutional animal ethical committee. They will review your proposal. Once if they convinced, then they can approve for animal studies. So what I'm trying to say is that even for doing the preclinical studies on animal, you cannot do that on your own. You need a very professional, ethical approval from institutional animal ethical committee. So it is not guaranteed that always your proposal will be accepted by the institutional animal ethical committee. More chances, there are chances. I cannot say more chances. Some chances for the proposal to get rejected. Many reasons there. I'm not going to discuss about that. So this is the overall idea of how to proceed with animal studies. Okay. So let's say that you got approval from institutional animal ethical committee, right? So you say you purchase the rat from your animal house. So in Shastra University also we have animal house. So near to the mechanical building, or near to in between the mechanical block and ECE triple block, you can see one building. Near to that parking space, there are some small canteens there. No, so there you can see one block. That block is called as animal house, right? That's a place where they breed the animal and they maintain the animal for the animal studies. So let's say you purchase the animal from animal house. So now you want to evaluate whether your drug molecule, say this is our drug molecule. We cannot say it as a drug molecule. Say the molecule which is promising on biochemical and cell line assay for type 2 diabetes. You have some molecules and you want to evaluate this molecule on animal studies. You purchase the animal, right? So what's the next thing is? The next thing is, normally, these animals, naturally they may have the type 2 diabetes sometimes, or you have to induce type 2 diabetes to the animals, because your intention is evaluating whether your molecule is promising for type 2 diabetes. So if I say that if the animal is if the rat is naturally having type 2 diabetes, it means that it has some genetic problem, and that problem lead for type 2 diabetes in that rat. So say if your rat is very healthy, if it don't have type 2 diabetes, what you have to do is that you have to induce the type 2 diabetes. So genetical models are very simple, right? So you just purchase the genetically present type 2 diabetic model. You just evaluate your drug molecule. It's a very straightforward method. But finding those kind of rats will be very difficult, right? So what normally the researcher in research institute do is they purchase the healthy animal and they induce the disease to the model. Okay, that's why it's called as animal model. We induce the diseased model. Okay, so there are different ways by which we can induce different disease to the animals. So there is a drug molecule called as streptozotocin. If you give that drug molecule for certain period of time to the rat or mice, what happens is that that will lead for development of diabetes. So after inducing the diabetes, we can ensure whether the disease is induced correctly using some parameters. So we should go for some assays or some biochemical parameters to ensure, to say that the animal is now having disease. Okay, that's the way how we have to induce the disease. So once if you induced the disease, or once if you have genetically modified diseased model, well and good, you can start your experiments, right? What we normally do is, you have to make a group. So this is group 1 animal and this is group 2 animal. So this is group 1 animal which are very healthy and they don't have any disease at all, and this is the type 2 diabetes with the help of drug molecule called as streptozotocin. Okay, so now you have to evaluate the effect of your drug molecule on the healthy animal model and the diseased animal model. So you have to start from some minimum dose, okay, and you have to increase the dose, and you have to check that till what dose the animal is very safe and there is no occurrence of death of the animal. Till that you have to increase, right? So in this procedure, at one point, at some concentration, we can say that the concentration may be toxic for the animal will start die. So that concentration is the upper concentration, right? So beyond that, if you go, the animals will start die. So based on that experiment, we can calculate what is the LD50, right? So what is the concentration that is required for killing 50% of the animal? This LD50 is called as a lethal dose 50, that we can measure by playing with or changing the concentration of the molecule that you're working on, right? So this is done with the normal animal, right? This is done with absolutely the normal animal. So now you take the drug molecule and go to group 2. The difference is group 2, they have now the disease, right? So now say the safety is studied using the healthy animal, and now you are going for the diseased animal, in which mainly you are going to study about the efficacy of the drug molecule. Okay, so what's the meaning of efficacy of the drug molecule? So these are type 2 diabetes animal model. So if you give some glucose, or if you give sugar rich, carbohydrate rich supplements or food to this animal, what happens is the blood glucose level of this animal models will be very high, because it's a type 2 diabetes, right? So obviously in type 2 patients, what happens is normally there will be a rise in the blood glucose level in the circulation, right? Glucose in the circulation will be very high. So once if you administer your drug molecule, if there is no change in the level of the glucose in the blood, which means that your molecule is not working on that animal model, your molecule is not having any efficacy. The other possibility is you are giving the drug molecule to the group 2 animals which are diseased. Okay, normally the blood glucose level in these animals are high after supplementing with some carbohydrate rich food. But once if you supplement your drug molecule after that, now if you measure the blood glucose level of that diseased model, the blood glucose level is, it's decreased to some extent, significantly decreased. What it means that the drug you have administered is having some efficacy. It is able to reduce the blood glucose level. That is the intention of your study. So now you can say that your molecule is having efficacy. It is able to reduce the blood glucose level. So those type of things what we normally study in the preclinical studies with the help of the animal models. So two things we study. I just wanted to summarize whatever I have discussed with you so far. So two things we have to ensure. The first one is, so what is the dose up to which the animals are safe? So in other words, what is the dose in animal which is very safe with minimum toxic effect? So this is what we study using the animal first. Second thing is, using the same animal, we study efficacy of the drug molecule. So two possibilities: you may end up with no efficacy, or you may end up with, yes, there is a kind of efficacy. So ability of the drug molecule to cure or minimize the disease. As of now, that's the definition for efficacy. So if your molecule is able to reduce the disease or control the disease, so at what concentration it does, that we can study with the help of animals. And this study is absolutely what is called as preclinical study. So along with that, the scientist will also perform a study about the pharmacokinetics of the drug molecule using the animal model itself. So they will study what is the absorption, distribution, metabolism, excretion of the drug molecule of your interest. All this is basically studied with the help of the animal model. I don't know whether my explanation is understandable. Any question I can address up to this. I hope I am connected. I hope that there is no time lag between what I speak and is there any time delay between? Huh?
No, sir.
Okay. Whatever I write, you are able to see immediately, right?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, fine. So I hope you understand, right? So this is this is what is called as a preclinical studies, right? So now I just wanted to tell a little more about the lead optimization, as I said earlier. Say for example, the molecule is having a very good efficacy. The molecule is having good absorption, distribution, metabolism, excretion, but it lacks the efficacy. The efficacy is not as expected. It's very less. So what the scientist will do is that they will take the original structure of your drug molecule and they will make small change at the level of functional group. And they will again evaluate the same molecule using the animal model. Now they will check whether the absorption is good, distribution is good, metabolism is good, excretion is good. They will also check whether the efficacy is increased or not after the change. Okay, so if it is increased, then it's successful, right? But whenever you make small change to the structure of the molecule you are working on, say for example, now the efficacy is increased, but the absorption is decreased. Okay, so again, problem for the medicinal chemist. Again, they have to make some change in the structure. So they will do this repeatedly again and again and again. It's an iterative process. So when they will stop this, they stop this until they got a molecule that is having a balanced property for absorption, distribution, metabolism, excretion, less toxicity, and good efficacy. Okay, that molecule we can say as a very balanced drug-like molecule. And those molecules are what are called as candidate drug molecule. I think first time I am defining candidate drug molecule. So what are candidate drug molecules? The candidate drug molecules are the one which are ready to evaluate in human system, which shows good absorption, distribution, metabolism, excretion with less toxicity and a desired efficacy on animal model. Those molecules should be called as candidate drug. Is the definition understandable?
Yes, sir.
Yes, okay. So I assume that everyone is following me. So what we have is a candidate drug molecule with us. Just now we have seen what is meant by candidate drug. Okay, so you're lucky that you got candidate drug molecule. Even at institutional level, we can identify good potential candidate drug molecule, no issues, right? So once if you have successful candidate drug molecules, then you should go for application called as IND enable. Okay, so IND stands for investigational new drug. Investigational new drug. I hope that my handwriting is somewhat readable, right? Is it readable?
Yes, sir.
Yes, sir.
At least to some
Yes,
extent,
sir.
right? Okay, so it's an investigational new drug, right? This, there is a authority called as FDA, stands for Food and Drug Administration. Food and Drug Administration. So this is the authority to which you have to submit your IND, investigational new drug. Okay, so what are the details you have to submit to this authority? So whatever experiments you have done with your biochemical assays, whatever experiments you have done with your cell line assays, okay, whatever experiments you have done with your preclinical, with the help of animal models like absorption, distribution, metabolism, excretion, toxicity studies, efficacy studies, all those details should be submitted to FDA through the application called as IND enable, investigational new drug. So they have very strong panel of members who are expertised in the disease you are working on. The variety of people will be there, right? So physicians will be there, medicinal chemist will be there, molecular biologist will be there, pharmacologist will be there, right? So it's a very big panel. So the panel will go through your application form. So they thoroughly go through what are the experiments you have done in biochemical assays, cell line, preclinical. They will go through thoroughly what are the details you are submitting related to the toxic studies, what type of toxic studies you have performed. So they will look for acute toxicity, they will look for subacute toxicity results, they will look for chronic toxicity results. All those they will look for. What are the experiments you performed to ensure that the molecule is having efficacy, right? So all those details will be gone through by the panel of members. So two criteria they will look, one is your data should convincing them that the molecule is very safe on animal. Second, your data should convince them that it is having very less toxic effect or no toxic effect. And thirdly, they will look for whether your molecule is having significant efficacy to control the disease on animals. So these are the three categories that they will look for in your application form. So once if your application is satisfied, once if maximum number of panel member accept the experiments you have performed to ensure that safety, toxicity and efficacy of the molecules on animal, then they may approve your application. So your investigational new drug application will be approved by FDA in case if you have very satisfactory data. There are chances also that they may reject. That depends on the data that you are submitting through the application. That is what is called as IND enable. So say for example, you are very lucky because your molecule is very safe and convinced the FDA through IND application. Now you are ready to go for a study called as clinical studies. Okay, so this is very difficult to carry out on institutions like universities or thing, right? So mostly this is done by pharma company. So this is a place where the pharma company enter into picture, because it's a time consuming process. Clinical studies need, we have to do this very carefully, right? And also it is a very time consuming process. You need a lot of money, right? So there are so many factors there in clinical studies. Also the difficulty of clinical studies are, they are very difficult, more complex comparing to the animal models, animal studies, right? So luckily you entered into the clinical studies. So clinical studies, we have different phases. So basically we have 3 phases: phase 1 clinical trial, phase 2 clinical trial, and phase 3 clinical trial. So there is one more clinical trial, okay, that is called as phase 4 clinical trial. So this 3 are first category of phases: phase 1, phase 2, and phase 3 clinical trial. So let me tell you what exactly they do in phase 1, phase 2, and phase 3 clinical trial. So I am going to tell you what normally the company, what they do in phase 1 clinical trial, right? So in phase 1 clinical trial, the companies like Pfizer or Novartis, there are so many companies. I am taking this because these are top, pharma companies in the world. Say Pfizer is having some successful molecule on animals. They wanted to evaluate on human, and they got approval from FDA through investigational new drug molecule. So now the Pfizer will call for volunteers. Okay, so they publish some information on the website. So they call for volunteers. So the volunteers they look for phase 1 clinical trials, they need a healthy volunteer. It's a very interesting thing. Okay, the drug molecule is designed for some disease, but in the phase 1 clinical trial, they look for some person who is very healthy, having no disease, any diseases, right? So those are called as healthy volunteers. So they call for the healthy volunteers. They will say that we have some drug molecule which is very promising on animal models, and we just wanted to evaluate on the human system. And so they need some person who are interested to intake this molecule and participate in this clinical trial, phase 1 clinical trial. Okay. So not all will show the willingness, right? So some may show willingness to the company, so they can submit the application form. So once if they have so this phase 1 clinical studies, the population size will be very small, so not more than 10 to 50. It's a maximum size we can say, right? So not more than that. So using 10 to 25 or 50 healthy people, they first evaluate the effect of the newly discovered molecule, which is having good efficacy on animal models. This is how they start, right? So the consent is very important. So they will get some legal documentations there. You have to sign on those legal documentation for participating in this clinical trial 1. Okay. So what I mean is that randomly they cannot give this molecule to the humans. So with the knowledge of the person, they have to give the molecule, right? So what they do in clinical trial exactly is, so once if they identify 10 to 50 healthy volunteers who are willing to participate in this phase 1 clinical trial of the drug molecule, so they start with very minimum amount of dose of the drug molecule, and they will give that to those persons who are interested, and they will start monitoring it. So initially they will start with a very minimum dose, because already they know what is the LD50 in animals. So there are some formulas to calculate what will be the safe dose for human. There's a kind of we can match. We have some procedure for translating the animal dose to human dose. So there are a lot of formulas which are involved. So they know what's the maximum upper limit we can go for human. So, but they won't initially go for that upper limit. So they start with very minimum concentration of the drug molecule. So for, say for example, they start with 1 mg or less than 1 mg of the molecule. They will give to all the healthy volunteers who are participating. So they will see, is there any undesired effect that is taking place in the normal healthy volunteers while giving the 1 mg. So they will check for the normal physiological function. So you know that there should be some standard value for the blood pressure. So your heart should beat at certain rate. So those are called as normal physiological function. So after giving this new molecule to the healthy volunteers, they will check whether the normal is there any change in the normal physiological function, or any undesired effect is reported by any of the participant in this clinical trial 1. So if nothing is observed, if everything goes fine, then slowly they will increase the concentration. They will now check, is there any change which is observed, and they will slightly increase that. So they will increase this till they find some undesired effect that is happening for certain people who are participating. So they may fix that range as the highest concentration. Okay, so now they know the minimum concentration. They cannot fix the minimum concentration now. So now they know what is the highest concentration till which there is no undesired effect, or there is no much changes in the normal physiology function of cellular events in the normal healthy people. So that is what they do in clinical phase trial 1. So once if you completed the clinical phase trial 1 without any reported toxic effect, severe undesired effect. So what if any undesired effect, severe undesired effect is observed, then they cannot go for phase 2 clinical trial. Okay, even if, say for example, even while giving the 1 mg minimum concentration of the newly discovered molecule to human, if any undesired effect, say for the example, the heartbeat is it's very high now, even after taking 1 mg of the molecule, definitely they cannot increase the concentration and they cannot go further, right? So what they have to do is they have to again go back for the lead optimization. They have to look for why the heartbeat is increased, or why the pressure, blood pressure is increased because of taking even minimum concentration. They have to address. They cannot directly take this molecule for phase 2 clinical trial. So once if no major undesired effect is observed, then you can go for phase 2 clinical trial. So let me tell what is the difference between phase 1 and phase 2 clinical trial. In case of phase 2 clinical trial, the population size, right? So as I said, 10 to 25 is a very small size. Now in phase 2 clinical trial, they may increase this to, say for example, some hundreds. So 100 to say maximum they can go for, the company will decide, right? So what's the sample size that is required? Not too much. Again, the sample size is slightly increased, but we cannot say this is a very large sample size. So now the company will again call for the volunteers. This time they call for diseased people. Okay, so not the healthy volunteers. So they call for diseased persons. So in the website, the Novartis or the Pfizer, they will publish that they got success in the newly discovered drug molecule on phase 1. Now they need some diseased volunteers to evaluate the newly discovered molecule. So this time they specifically look for diseased person for which for the disease for which the drug molecule is discovered. Say as we took that molecule is discovered for type 2 diabetes. So what they do is they specifically call for type 2 diabetic patients for participating in clinical trial 2. So once if enough number of volunteers they got, with what they can start the clinical trial 2. So first they will evaluate the person. They will ensure that the persons are having the participants are having exactly type 2 diabetes, because we have different types of diabetes. The molecule is discovered for type 2 diabetes. First they will evaluate whether the patients are having only they're having specifically type 2 diabetes. Secondly, they will ensure that the patients are not having any other disease, comorbidity, right? So we call it as comorbidity. So the company has to ensure that they don't have any comorbidities, right? So sometimes the type 2 diabetes patient also having some thyroid, or he may have some heart related diseases, right? So for phase 2 clinical trial, those type of patients are not preferred. The patients preferred for phase 2 clinical trial is absolutely they should have only the disease for which the drug molecule is discovered, not having any other comorbidities or any other diseases. Is it understandable?
Yes, sir.
Yes, sir.
Okay. So now what they study in phase 2 clinical trial is, again they will check for dose, because the dose which is safe in the normal person may not, there may be slight changes in the metabolic pathways and functioning of organ system in the diseased patients. So again they will go for fixing the dose molecule. So initially they will start with very less dose, as I said in clinical trial 1. Slowly they will increase the dose, and they will look for the effect of increasing the concentration. Parallelly, what they do is, this is very important, they will check for efficacy of the drug molecule. Very important. So efficacy of the drug molecule is same thing what I explained for preclinical studies, the ability of the drug molecule to cure the disease. Okay, because this time the volunteers are having the disease for which the drug molecule is discovered, right? So what happens in type 2 diabetes, as we said earlier, there will be always increase in the concentration of the blood glucose level in the circulation whenever you measure, right? So there will be higher concentration of the blood glucose that may affect the organ. A lot of complexities it may cause, right? So what they will check is that whether after taking the drug molecule, okay, so at what concentration they're able to they're able to see there is a kind of reduction in the blood glucose level, that they will measure. Unfortunately, if your drug molecule, even at the higher concentration, is not able to decrease the blood glucose level, okay, your molecule got failed. That's what we can say, right? But your molecule is promising on animal, not necessarily that the same thing should happen in human, because we cannot compare the animal models with human, because the humans are we have very complex regulatory mechanism that we cannot expect in the animal models in which we are working on. Animal models are just model, a mimic of the human, not exactly, to some extent. So if your molecule is having efficacy and no undesired effect is identified in this set of population, then you are very lucky that you can take this molecule further for phase 3 clinical trial. So I can say that most of the drug molecules will get failed only in phase 2 clinical trial. And the main reason for the failure of the drug molecule at phase 2 clinical trial is because of lack of efficacy. What I mean here is, the molecules which is promising on animal most of the time may not be promising on human. So what they do in phase 2 clinical trial, if we summarize, so once again they try to fix the dose of the newly discovered molecule, to what extent, to till what dose it is very safe. First thing. Second thing, absolutely very important for any drug molecule, is the efficacy of the drug molecule. The drug molecule could able to cure the disease for which the molecule is discovered. If the molecule is not doing that, we can say that the company is completely failed. Is it understandable?
Yes, sir.
Yes, sir.
Okay, now moving on to phase 3 clinical trial, let me tell what's the major difference between phase 2 and phase 3, because we already know what's the difference between phase 1 and phase 2. So the population size in phase 3 is highly increased. Maybe they may call for 500 to 1,000 or even more than that as volunteers. So now this time it is across the world. Till phase 2 trial, it's given for small population. Okay, if the molecule is discovered in India, so maybe it is tested with some Indians, not with other foreign country people. They may add 1 or 2, but not too much, right? So but in case of phase 3, now the molecule will be given and evaluated. The volunteers from different part of the country will be called for participating in phase 3 clinical trial. As the first difference, the population size is large, and the people are now from different different countries. Why I am emphasizing this is, different the genetic content of the people from different part of the world will differ. If you compare the genome of ours with the Americans, there will be definitely some kind of variation. If you compare our genome with Japanese, there will be kind of difference. So those people will have some type of genes and will be having some type of genome. There will be slight variation across genome, right? So now your molecule is promising on a certain group of people. Now the big question is, if I give that to people with variety of genetic variation, the molecule should be promising. That is what is evaluated in phase 3 clinical trial. So across the world, they pick up the volunteers. Now also the volunteers are not healthy volunteers. Now the volunteers are just like how they pick up in phase 2 clinical trial, diseased volunteers. So now in phase 2 clinical trial, the participant should have only that specific disease. But in phase 3 clinical trial, they will call for the people with comorbidities. Okay. So because, there will be always a cross communication between different diseases, say thyroid, say diabetes, say heart disease. A person is having 3 diseases and there will be a crosstalk between these 3 diseases, right? So what's the question is that the molecule which is promising on the patients having only type 2 diabetes, is it going to work for patient having multiple diseases or more than one disease, which is having cross communication? That question they have to address in phase 3 clinical trial. So they will call for the patients with comorbidities. Understandable? So now again, they will give the drug molecule and they will evaluate. The molecule should be promising this situation also. So once, if you are lucky enough and if your molecule is very promising, even across the people across the world and having different diseases, multiple diseases, then your company is really lucky, right? So because the molecule is not producing any undesired effect, no much toxic effect is shown in any people, and the molecule is highly having efficacy with different people with different genomic conditions across the world. And even your molecule is very promising with the patients having comorbidities. So you are very lucky, right? So now you have huge amount of data from phase 1 clinical trial, and you have huge amount of data from phase 2 clinical trial. You have huge amount of data from phase 3 clinical trial. So all these experimental details and results you have to share with the FDA again, United States Food and Drug Administration, through an application form called as NDA. And NDA stands for New Drug Application. It stands for New Drug Application. So through this NDA, you have to submit all the details. Okay. So there is a saying, right? So by one of the medicinal chemists, right? The amount of data submitted to FDA through NDA is like, Eiffel Tower, right? So if you stack the data submitted to the FDA through NDA, if you stack the paper, right? So the height of the paper will exceed the height of the Eiffel Tower. So that's how that much amount of data has to be submitted to the FDA through NDA application. So the FDA will go through the application. So as I said, they have very strong panel members. They will go through all your data. So they may take some time to go through the entire process, entire results that you have submitted, because you're going to submit huge amount of data, right? So once if the panel members are convinced, once if the FDA overall convinced with your data, if they feel that the molecule is highly safe in human and it is able to produce the desired biological effect, it's having efficacy, then they may approve your molecule. Okay. So once if your NDA is approved, then your drug-like molecule, candidate drug molecule, now can be called as drug molecule. So these are the various, you can market this as a drug molecule. So you are the owner of this molecule. No other company can market this as a drug molecule because you discovered this. This molecule will be subjected for patent procedure. Okay, so you will be given certain time, maybe some 7 to 10 years. No other companies can manufacture the same molecule. You can manufacture, you can sell. So whatever amount you spent on this project, you can make from the income, you can make profit of this molecule. So these are the various stages of modern drug discovery process. Is it understandable?
Yes, sir.
Any question I can address? If not, I think time up. We can wind up. So anything learned?
So what will be in phase 4, sir?
Phase 1, phase 4, I think time is over. Okay, let me finish, right? So maybe another 1 or 2 minutes, let me finish. So phase 4 trial is, after marketing, right? So the molecule, say for example, you marketed the molecule as a drug molecule. You named it with something, right? So say aspirin is marketed, right? So what now the physicians, the physicians, they will came to know about this molecule. What for this molecule is discovered. All the doctors will be, the representative, you can see the company representative will go meet the doctors, and they will tell more details about the newly discovered drug molecule which got approved and marketed. And once if the physicians are willing to prescribe this, they can prescribe this molecule to the patients who are coming to them. So what the physician should do is, if the molecule is very newly discovered molecule, this molecule is subjected for monitoring. So who monitor this? The physicians, the doctors will monitor this molecule. So whenever they prescribe this newly discovered molecule to any patient, they carefully monitor the effect of this molecule on patient. The reason is, once if you market, right, so the population size is now huge. Whoever is having the disease, they are going to take this drug molecule for as a treatment procedure, right? Population size is very large, so the physician should monitor. Okay, so up to phase 3, the population size is very less. The molecule is very safe. Okay, it is not necessary that the molecule should be safe. If we increase the population further, maybe chances some of them may have encountered with certain undesired effect or very toxic effect. Say for example, a group of people report that the molecule is having some toxic effect. So this can be observed only with the help of physician. The physician monitor this. They whenever any undesired effect is reported because of taking this newly discovered molecule, that will be informed to FDA by the physician directly. So the FDA will go through whatever is detailed by the physician. And if the same issue is raised by many physicians, what the FDA will do is that the FDA will ask the company to withdraw the drug molecule from the market because of so and so reasons, so and so toxic effects which are reported by physician. So without any question, the company has to withdraw the drug molecule from the market, and that stage is what is called as phase 4 clinical trial. So this monitoring is maybe for 1 or 2 years, or 6 months to 2 years. A minimum time they will monitor the effect of this drug molecule. Is it understandable?
Yes, sir.
I can say that there are many molecules which are withdrawn at phase 4 trial, which means that after marketing the drug molecule and because of some undesired side effects which are reported by physicians. I think I completed phase 4 also. Is it fine?
Yes, sir.
Anything have you people understand from the drug discovery process? Is the concept clear?
Yes, sir.
Yes, sir.
Okay. I think we shall wind up the session. Okay. Thank you.
Thank you, sir.